Good afternoon, everyone. I mean, it's afternoon for me. So, anyway, welcome everyone. This is the second lesson that I'm going to teach. I thought it was a very important topic, so I wanted to discuss it towards the beginning. It's cell cycle and cell division. And before I begin the topic, I would like to ask you all to kindly share the video as much as possible, like, share, and subscribe, because this is my second video itself. And to be very honest, I want to reach as many people, as many students as possible, because I think it will be beneficial for not only on Neet UG, although that's the point of view from which I'm teaching, but also for your other entrance examinations, be it now or in the future. So, if possible, kindly share the video. About myself, my name is Ojasvi, and these are some of the qualifications that I have and some of the examinations that I've cleared. With this, let's just start directly. So, our topic for the day is cell cycle and cell division. I'm going to read the paragraphs, and whenever I find something that is important, I will stop and explain that. Most of the questions that have come in the past few years are directly from mitosis and meiosis, specifically the stages. Like they will ask you that if the genetic content at the beginning is 2n, what will be the genetic content in G2, or what will be the number of chromosomes in meiosis 2's anaphase. So those sort of questions come. So till we reach that point, let's go to the chapter and let's see what NCERT has given us and how to build up your important information notes from it. Okay. So are you aware that all organisms, even the largest, start their life from a single cell? We all know that, right? You may wonder how a single cell then goes to form such large organisms. Growth and reproduction are characteristics of cells, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells. We all know that, right? That from a single cell, there is a cell cycle or mitosis giving rise to two cells. And in case of meiosis, what happens that there is 2n, it gets divided into 4 cells of n, n, n and n each, right? Let's move ahead. So let's go to the cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also takes place. All these processes, that is cell division, DNA replication and cell growth hence have to take place in a coordinated way to ensure correct division. Exactly, right. So for example, if there is no DNA replication, but the cell is growing and dividing, so what will happen instead of the daughter cells getting 2n and 2n, if there is no duplication of the DNA, which happens in the S phase, right? So what happens in S phase, your genetic content doubles. For example, if the initial genetic content was 2n, in S phase, this 2n becomes 4n, right? So if this does not happen and still cell division continues, then the daughter cells would have n and n, which is the haploid content, and then that organism, those cells will not be able to survive, right? So, for proper division, you need replication. So, DNA replication and cell growth hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes other constituents of cell, and eventually divides is known as cell cycle. We all know that, right? Although cell growth is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosomes are then distributed by complex series of events occurring during cell division. Alright, so let's move ahead. This is your cell cycle. This is the S phase or the synthesis phase. In the synthesis phase, your genetic content is doubled, right? So if I have to formulate sentences, in your G1, your number of chromosomes would be 2n and your genetic content would also be 2n. In your S or synthesis phase, what happens that your chromosomes are still 2n, but the genetic content gets doubled and it forms 4n, right? Because it is synthesis, right? And in G2 also the same thing. Mm, it's the same number of chromosomes, the same number of genetic material, that is 4n and 2n. Alright, and after that you have your mitosis phase, right? So if you can see, majority part of your cell cycle is taken up by S phase, right? So synthesis phase takes the most amount of time. Now, the cells divide once in approximately every 24 hours. We are talking about a typical eukaryotic cell over here. So a typical eukaryotic cell takes around 24 hours. When you're talking about bacterial cells, it takes, as you may know, on 16 to 20 minutes depending upon the bacteria. So these cells divide once in approximately every 24 hours. However, this duration can vary from organism to organism, also from cell type to cell type. Yeast, for example, can progress through cell cycle in only 90 minutes because yeast is it's a unicellular eukaryote, right? In humans, which are complex eukaryotes, it takes around 24 hours. So the cell cycle is divided into two basic phases. One is interphase. Interphase consists of your G1, S and G2, and then M phase or mitotic phase. So the M phase represents the phase when the actual division or mitosis occurs. So over here only division is occurring. The part where it is DNA replication part occurs in your S phase. After it has replicated, then the division occurs in your M phase. Okay. It is significant to note that the 24-hour average cell division proper lasts only for about 1 hour. That means your M phase only lasts for about 1 hour, right? The interphase lasts more than 95% of the duration of the cell cycle. That means G1, S and G2 take about 95% of the total time. That is 95% of 24 hours goes into your interphase. Let me just write it down. All right. So the M phase starts with nuclear division. So what happens, if you remember, in the cell structure, there is the nucleus, and inside the nucleus you have your chromosomes, right? So for the chromosomes to get divided equally between two cells, the first thing that happens is the breakdown of your uh, nuclear membrane. Okay, so the M phase starts with nuclear division corresponding to the separation of daughter chromosomes, which is known as karyokinesis, and usually ends with division of cytoplasm, which is known as cytokinesis. So when you're talking about, for example, this is a chromosome, these are chromosomes. So when the chromosomes will be dividing, right? So the division of chromosomes, so that each daughter would have one chromosome, or n number, I mean half of the 4n, that is 2n. Each daughter cell has 2n of chromosomes. This step is known as karyokinesis. Okay, and when you're talking about the division of cytoplasm, that means the cell, once the cell has equal number of chromosomes, so the cell starts dividing, right? Like this. This division of cytoplasm is known as cytokinesis. Okay, so don't get confused between the two terms. Till now, in Neet UG particularly, questions have not come as in karyokinesis and cytokinesis or anything regarding that. But it's important to clear this for your basic concepts. The interphase, though called the resting phase, is the time during which the cell is preparing for division. So, the cell has to prepare excess of everything, right? Since from one cell you have to make two different cells, so you need that much amount of stuff to share between the two, right? So in the synthesis phase, the proteins, the nucleotides, all these are synthesized so that they can equally get divided between the two daughter cells. Okay. uh, DNA replication in an orderly manner, the interphase is divided to further steps. Alright, now let us move on to the more important topics from where questions have come. So it's basically your information about G1, S and G2 phase along with the different stages of mitosis. Since today's lesson's topic is mitosis, so I'll be focusing mostly on that, and in our next session we can focus on meiosis as well, right? So let's talk about G1, S and G2 phase. First, let's read and let us try to analyze what the statements are saying and make a roadmap out of it, right? So G1 phase corresponds to the interval between mitosis and initiation of DNA replication. So G1 phase. During G1 phase, the cell is metabolically active and continuously grows. So the purpose of G1 phase is growth of the cell but does not replicate its DNA. So if the initial content of the cell was the chromosomes were 2n, that is diploid cell, the DNA content is also 2n, alright, but does not replicate its DNA. S or synthesis phase marks the period during which DNA synthesis or replication takes place. So S is where DNA synthesis takes place, DNA replication. Alright, so your number of chromosomes would be 2n and the DNA content would be, since DNA replication has occurred, so the 2n amount of DNA has now become double. So it would be 4n now. Okay. So replication takes place during this time. The amount of DNA per cell doubles. If the initial amount of DNA is 2C, then it increases to 4C as we have currently marked. However, there is no increase in the chromosome number, as I have said over here. There is no increase in the number of chromosomes. If the initial number of chromosomes was 2n, even after S phase, the number of chromosomes would be 2n. Alright. However, there is no increase in number of chromosomes. If the cell had diploid or 2n number of chromosomes at G1, even after S phase, the number of chromosomes remain 2n. In animal cells, during the S phase, DNA replication begins in the nucleus. Alright. So obviously we all know that DNA replications with the help of DNA polymerase takes place inside the nucleus, right? DNA replications begins in the nucleus and the centrioles also duplicate in the cytoplasm. During the G2 phase, proteins are synthesized. So in the G2 phase, protein synthesis occurs and the number of chromosomes still remain as 2n. The DNA content, just like its previous step, is still 4n. Alright. Proteins are synthesized because only proteins are synthesized, right? If the genes were synthesized, then it could have changed from 4n, but since it is synthesizing only the proteins, the number of chromosomes and the genetic content remains constant. Okay, some cells in the adult animals do not appear to exhibit division like your heart cells. Questions have not come in Neet from this particular statement, but this statement is important from the point of view of other entrance tests like your QET, etc. And many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells that do not divide further exit the G1 to enter quiescent stage. Now, when you're talking about quiescent stage, it is a stage in which the cell is still alive, it is still breathing, it is still consuming energy and food. So, the cell is metabolically active. Okay. However, it is just not choosing to divide. It is choosing to live, but it is choosing to not divide at this particular stage. So they enter an inactive stage called quiescent stage of the cell cycle. Cells in this stage remain metabolically active, but no longer proliferate unless called on to do so, depending on the requirement. In animals, mitotic cell division is only seen in diploid somatic cells. So when you're talking about animals, over here, you will only see mitosis in diploid cells. However, in case of plants, you will see mitosis in both diploid cells as well as haploid cells. So this is my homework to you to solve this particular statement. From your recollection of examples of alternations of generation in plants, specifically your Bryophyta, Pteridophyta, identify plant species and stages at which mitosis is seen in haploid cells. This is the question that I'm giving you, and whoever watches this lesson and is studying hard for Neet, It would be really nice if you all can write the answer in the comment. All right, now let us try to solve this particular question. How do plants and animals continue to grow? So over here it's growth. So we are talking about mitosis. Do all cells in a plant divide all the time? So no, not all the cells divide. There are some cells which are continuously dividing, like your apical meristems. Okay. Do you think all cells continue to divide in all plants and animals? No, all cells are not dividing in all plants and animals. When we're talking about animals, you can give an example of stem cells. Uh, but then there are also cells which have stopped dividing, like the skin, right? And then in plants, meristematic tissue, apical meristem, specifically when you are talking about shoot apical meristem and root apical meristem, they are dividing, but fully differentiated cells like the cells of your leaf, once they have gained their function, they do not divide, right? Can you tell the name of the location of the tissues having cells that divide all their life in higher plants? I've already told you apical meristem. Do animals have similar meristematic tissues? So animals do not have meristematic tissues, but animals have pluripotent stem cells and totipotent cells, right? So then again, this is another homework. If you're talking about plants, you call them meristematic cells. If you're talking about animals, you call them pluripotent and totipotent cells. So firstly, find out the difference between pluripotent and totipotent, and then find out which cells in an animal are known are examples of pluripotent cells and which cells are examples of totipotent cells. All right. Then you have studied mitosis on onion root tip cells. It has 16 chromosomes in each cell. How can you tell how many chromosomes will the cell have at G1 phase, S phase, and after M phase? So number of chromosomes. You will get the answer from this particular information that I've given. All right. Also, what will be the DNA content of the cells? Again, you will get the information from this particular piece of information that I gave you. In case it is still not clear to you, you can write it in the comment. However, I think it is pretty much, it's the same thing. It's literally the same thing. However, if you still have any issues, I am open to explaining it again. So yeah, just you can comment. Let's move forward. So this is the most dramatic period of the cell cycle involving major reorganization of virtually all components of the cell, since the number of chromosomes in the parent and progeny cells is the same, right? 2n mitosis. Again, you are getting two daughter cells and each daughter cell has 2n and 2n. All right. So it is also known as equational division. That means equal, everyone is getting the equal amount of chromosomes as the parent. Okay. Though for convenience, mitosis has been divided into 4 stages of nuclear division. It is also very essential to understand that cell division is a progressive process and very clear cut lines cannot be drawn. So when you're actually visualizing cell division, you'll be able to visualize that, okay, the cell is now in the metaphase, all the chromosomes are aligned at the, equator. But when you're visualizing it, clear cut lines cannot be drawn, like, okay, so prophase has just ended and metaphase has just started. You will not be able to draw a clear cut line. It will be like, okay, you'll be able to see the chromosomes if they are now in prophase, and eventually after some time they will be in metaphase. So that's how it goes. Okay, now let's talk about prophase. And these are the stages. You should know the stages on your fingertips, like pro, meta, ana, telo. Okay, so that's how I also remembered pro, meta, ana, telo, pro, meta, ana, telo. If you repeat it like 10 times, you'll eventually learn it, pro, meta, ana, telo. All right. So let's talk about prophase. In prophase, the major thing that happens is condensation of your chromatin fibers. So prophase, which is the first phase of mitosis, follows the S and G2 phase, right? After G1 comes S, after S comes G2, after G2 comes prophase. So in the S and G2 phase, the new DNA molecules formed are not distinct but intertwined. So after replication, you'll find that, okay, new DNA content has been generated, etc. but they are intertwined. Prophase is marked by the initiation of condensation of the chromosomal material. So this whole genetic content starts condensing, and eventually it forms condensed chromosomes. You'll be able to clearly see. In this stage, you will not be able to see the genetic material. In prophase, since the genetic material has condensed so much, now you will be able to clearly visualize it. Okay. The chromosomal material becomes untangled during the process of chromatin condensation. The centriole, which has undergone duplication during S phase of interphase, now begins to move towards the opposite poles of the cells. So what happens? This is your nucleus. If this was the centriole, during the S phase, the centriole also duplicates, right? This is during the S phase, and this is your genetic material. In the prophase, what happens? This is your nucleus. This is your genetic material which has become condensed, and over here the centrioles will start moving towards the pole. All right. Okay. Now begins to move towards opposites of the pole. The completion of prophase can be marked by the following characteristic event. So the chromosome material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids attached together at the centromere. So this part is known as the centromere, the part where they are attached to each other. Initiation of the assembly of mitotic spindle, the microtubules. Right, so the centromeres are over here. From the centromere starts extending microtubules and other components of your cytoskeleton, right? So initiation of mitotic spindle, the microtubules, the proteinaceous component of cell cytoplasm helps in the process. All right, let's move ahead. So cells at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, and the nuclear envelope. So this is important for your own understanding, that when the cell has entered cell division, in that stage you will not be able to visualize the components like Golgi, the double membrane compartment, endoplasmic reticulum, again double membrane, nucleolus, and the nuclear envelope. Alright, obviously nucleolus and nuclear envelope you will not be able to visualize because they disintegrate. They disintegrate so that the chromosomes can get attached to the microtubules. The microtubules are attached to the centrioles, right? If I draw this, this is the thing. These are your centrioles over here. These are your centrioles. These are your microtubules or spin. So spindle fibers are basically composed of microtubules. The spindle fibers, right? And if you look closely, so over here if you see, there's a structure, right? This is your chromosome, this is your centromere. The centromere is encased in a protein known as kinetochore, and this is the kinetochore to which your spindle fibers attach, right? Remember this information. Okay, now let's move to metaphase. So the complete disintegration of nuclear envelope marks the start of metaphase, right? So the mark of metaphase is complete nuclear envelope disintegration. This statement is important from the point of view of Neet. Okay, hence the chromosomes are spread through the cytoplasm. By this stage, condensation of chromosomes is complete and they can be observed clearly under the microscope. This statement has come many a times, which is the stage of cell division in which you can clearly visualize the chromosomes. So it is the metaphase. All right. This then is the stage at which morphology of chromosomes is most easily studied. At this stage, metaphase chromosomes is made up of two sister chromatids which are held together by the centromere. So two sister chromatids, these are your sister chromatids. This is one sister, this is the second sister. Together they are known as sister chromatids. They are held together by the centromere. The centromere is encased by kinetochore. Okay, small disc-shaped structure at the surface of the centromere are known as kinetochores. These structures serve as the site for attachment of spindle fibers to the chromosome that are moved into position at the center of the cells. Hence, the metaphase is characterized by all chromosomes coming to lie at the equator. So if you can see, this is the metaphase, and all of the chromosomes are very neatly arranged at the equator. From on each side of the kinetochore is attached a spindle fiber. The spindle fiber is then in turn attached to the centromere, and that's when the pulling will start in anaphase. So for the pulling to start, it has to first attach, right? So the attachment process and the what can I say, the geometrically aligned stage of the chromosomes happen at metaphase. The stage in which you can clearly visualize the chromosomes happens in metaphase, right? So the plane of alignment of the chromosomes at metaphase is refers to as metaphase plate. The key features of metaphase are the spindle fibers attached to kinetochores, and chromosomes move to spindle equator and get aligned uh, along the metaphase plate through spindle fibers to both the poles. Next comes anaphase. Anaphase is basically characterized by the stage in which the sister chromatids get separated from each other and get pulled towards one of the poles, either this pole or this pole. Okay, so at the onset of anaphase, each chromosome is arranged at the metaphase plate and it is split simultaneously, and the two daughter chromatids are now referred to as chromosomes of the future daughter nucleus. Begin their migration towards the opposite poles. As each chromosome moves from the equator plate, the centromere of each chromosome is towards the pole. So they start getting pulled by the, because the spindle fibers were attached to the kinetochore. The kinetochore is attached to the centromere. So if you are getting pulled, if someone comes and pulls you from behind, so what will be your structure like? Won't it be like this? Your upper body would be like this, your lower body would be like this, and the person holding you would be pulling from your waist area, right? So similarly, that sort of structure is formed, right? As each chromosome moves away, the centromere is towards the pole and hence the leading edge with the arms trailing behind. Okay, these are the arms, they are trailing behind. This anaphase is characterized by centromere splitting and chromatid separation, and chromatids move to the opposite poles. Now we are talking about telophase. So this is basically what you can say the last stage, because now they are clearly at opposite poles, right? So at the beginning of the final stage of mitosis, telophase, final stage, right? Final stage of mitosis, telophase, the chromosomes that have reached their respective poles decondense. Now the chromosomes have already reached their respective poles, so now they can start, sorry, I've messed this up, just give me a second, yeah. So now they can start decondensing, right? They will start decondensing because they've already reached their final destination. Okay, the individual chromosomes can no longer be seen since they start, detangling. Now they cannot be seen anymore like it was there in the initial, at the very beginning. They were chromatins, so they could not be seen. They condensed and became chromosomes, and then they could be visualized. Now they are returning back from the chromosome stage to the chromatin stage. This is the stage which shows the following key events. Chromosomes cluster at opposite spindle poles and their identity is lost. Now they are clustering towards one end and their identity is lost, right? Nuclear envelope assembles, and then nuclear envelope starts assembling around the chromosomes, right? Uh, so nuclear envelope assembles, nucleolus, Golgi complex, and ER reform. So we saw at prophase that these organelles were not visible anymore. But now that the division has occurred, now these organelles also start reforming, and hence they start forming their proper structures and can be visualized now at telophase, right? Now let's move to the end, which is cytokinesis. So mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, but the cell itself is divided into two daughter cells by a separate process known as cytokinesis. So till this point only karyokinesis occurs. Karyokinesis, that is the division of the nucleus, but it is still one big cell. Now this cell has to divide into two daughter cells. So in an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens as you can see over here, the furrow gradually deepens and ultimately joins the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall. Hence, over here, cell plate formation occurs in case of your plants. Okay. Undergo cytokinesis by a different mechanism. In plant cells, the wall formation starts in the center. Yeah. So one more thing, in your animal cells, this is how the furrow is extending from outside to inside. In case of your plants, what happens? The plate is formed over here, and then the plate starts growing like this. So in your animals, it is like this, and in your plants, the growth is like this. All right. Grow outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of a simple precursor known as cell plate that represents the middle lamella. So, in case of your plants, this is how it's arranged, the cell wall, right? Connecting two plant cells is your middle lamella, right? Represents the cell plate rather represents the middle lamella between the two vertices. At the time of cytoplasmic division, organelles like mitochondria and plasters get distributed between the two cells. In some organisms, karyokinesis is not followed by cytokinesis, and as a result of which multinucleate condition occurs. For example, the formation of syncytium and liquid endosperm of coconut. So what happens is that if the cell does not, if the cytoplasm does not divide, what would happen that this particular, for example, this particular structure, now this structure will have how many chromosomes? It will have 4n number of chromosomes, right? So it has, and two nucleus as well. So now it is a binucleate stage. Now, for example, if this particular big cell again undergoes replication but does not undergo proper cytoplasmic division, that is your cytokinesis, what will happen that these two nucleus will now become 4 nucleus and then 8 nucleus and so on. So it will become multinucleate cell. So now we'll focus on what is the significance of mitosis. So mitosis or equational division is usually restricted to diploid cells in animals. In case of animals, in plants it can happen in both diploid as well as haploid. However, in some lower plants and in some social insects, haploid cells also divide by mitosis. It is very essential to understand the significance of this division. Are you aware of some examples where you have studied about haploid and diploid insects? Have you? If you have, then comment the answer in the comment box. Alright, mitosis usually results in the production of diploid daughter cells with identical genetic complement. This is the major function. Identical genetic component. When you're talking about meiosis, over there you will study about recombination of, recombination of genes. So whenever the resulting, daughter cell would come, it will first be haploid. Then there would be a what can I say, in the reproduction process, there would be fusion of the sperm and the ovum. Then again, there would be different characteristics coming from both the parents. So basically, your genetic identity would be lost in case of meiosis. That does not happen in mitosis. In mitosis, the genetic complement is identical to the parent. The growth of multinuclear organism is due to mitosis. This is the second point. Cell growth results in distributing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the, this is the third thing, restoration of nucleocytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cells of the upper layer of the epidermis, cells of the lining of the gut and blood cells are being constantly replaced. Mitotic division in the meristematic tissue, etc., etc. All right. So we'll be studying meiosis in the next lesson. This was it from my side. If you have any questions, you can ask in the comment. You can also reach me at my email ID, which I'll be giving in the description box. There are a lot of questions that I want to discuss that have come in the last 10 years, and we'll be discussing all of the questions in the past 10 years in a separate video. First, I'll be completing your meiosis and I'll be giving you some important notes from where questions have frequently come. So that particular set of notes is very important for you. And after completing meiosis and those notes, then we will be moving to the questions. So there are around like 40 to 45 questions that I want to discuss. So I have to make a separate video for that so I can discuss the question in details. Yeah, so that's it from my side. I will try to upload those. First I have to make those videos. So I try to make those videos within this weekend. For that, again, I would like to request you to subscribe so that whenever I upload those videos, you will get the notification, and I'm sure it would be helpful for you. In case I have made any mistakes anywhere in the lesson, please feel free to correct me because I may be wrong as well. I am also learning. And in case you need any help in any particular topic, please reach out. Alright, so that's it from my side. Thank you for watching. I hope you all had a great time. Okay, bye.